Hello biology students, and welcome to our next unit, which is going to be all about biotechnology, which is DNA technology and genetic engineering. So some things you can do with DNA technology and genetic engineering include things like forensic analysis, CSI, solving the mystery crime scene kind of stuff. We can produce human proteins in mass in the labs. We can make transgenic organisms. We can do RFLP analysis, a whole bunch of stuff that we're going to talk about in this unit. So in this unit, there are hundreds and hundreds of different things you can do with genetic engineering and DNA technology. In this unit, we're just going to talk about 5, but I want you to keep in mind that there are a lot of cool applications that we just don't have time to talk about. So specifically, we're going to start with the production of human proteins. We're going to talk about how you could use DNA technology to identify people, like through paternity testing or through like CSI-style forensic analysis. We're going to talk about how you can use it to identify human diseases, like who has a disease and who doesn't. We can use it to track all human genes and also to make modified organisms like genetically engineered food. So we're going to talk about these 5, but remember there are lots of other applications as well. Okay, so we're going to talk in this video about the first half of the first use, which is producing human proteins in bacteria. And the example we're going to use is curing dwarfism or pituitary dwarfism. So basically, we're going to grow up the human protein for growth in bacteria and then give that hormone, that protein, to people who are dwarves and hopefully help to mitigate some of their symptoms. Let's look a little bit about background information about dwarfism first, because that's going to be our example for this section of DNA technology unit. So dwarfism is a recessive disease that causes adults to be no more than 4 feet tall. And these little people, the reason they're so small or so short is because they do not produce the growth hormone, that protein. So the gene they have for the growth hormone protein is mutated. So they don't make it properly. So more specifically, the gene they have that's more mutated is the GH1 or growth hormone 1 gene. And because they don't have this gene, their pituitary gland does not produce growth hormone, so they do not grow very tall. So there were a lot of early attempts to treat dwarfism. So, for example, in the past, people who were diabetic, doctors had realized they could just take the insulin from pigs and give the insulin from pigs to humans, and that kind of helped to treat diabetes. However, so they tried the same thing with dwarfism. They said, okay, I'm going to take the growth hormone from pigs and give it to people who have dwarfism and to see if it would work. But really, it did not work at all. And then they said, okay, so it has to be human growth hormone. So they tried taking the growth hormone from human cadavers, human dead bodies, but they found out it took 20,000 pituitary glands from 20,000 dead bodies to get enough growth hormone to treat one person. So both of these techniques really weren't working. On top of that, all these pituitaries from these cadavers, some of them were contaminated with prions, which are these nasty protein. They're like viruses, but they're made of protein, and which can cause brain disorders. So overall, all of their previous attempts really weren't working out for them. So then, finally, with the advent of DNA technology, scientists realized that what we can do is basically take the gene for human growth hormone, put it inside bacteria, and allow a bacteria to make us some human growth hormone. So this is exactly what we did. So by doing that, by putting the human gene for growth hormone into bacteria, what they were creating was called recombinant DNA. So a recombinant DNA is a single DNA molecule that results when scientists combine DNA from different sources. So here we were taking human growth hormone and combining it with bacterial DNA. So that was an example of a recombinant DNA molecule. So some other examples that are really common, putting human DNA into mice or putting mice DNA into bacteria. These are all examples of recombinant DNA technology. Okay, so how is this done? So here's maybe a human cell of a healthy person who does not have dwarfism. What we would do is we would take out the copy of their gene for normal human growth hormone. So this is a DNA molecule. We would then cut it with an enzyme, so the ends of the DNA molecule were sticky. In other words, they were single-stranded, so they're going to want to bind with another single-stranded DNA molecule. At the same time, we take a DNA molecule from bacteria called a plasmid. You can kind of think of this as a mini chromosome that bacteria carry around. We can take that bacterial DNA molecule, we can cut it open so it also has a single-stranded end, and then we can combine it. We can take this DNA piece, stick it in my plasmid, and then now I have bacterial DNA combined with human DNA. And then I can put this whole thing back into the bacteria and let the bacteria grow lots of copies of the human growth hormone gene for us. So let's look at another picture that's a little bit more complicated, but it's basically showing the same thing. So again, remember this plasmid, this is a small extra chromosome found in bacteria. So we take that plasmid out. We take the gene that I'm interested in out of a healthy person who has a normal copy of human growth hormone. I cut them so they have single-stranded ends that are going to want to combine into this recombinant DNA molecule. And then I put that back into the bacteria. Now remember, bacteria divide a lot, and they divide really fast. So if I let this bacteria grow for, honestly, like 24 hours, now I'm going to have millions and millions of copies of my bacteria. And that bacteria is going to have millions and millions of copies of that human growth hormone gene. So at this point, I could either remove all those copies of the gene, so now I have lots of copies to work with, or I could just let the bacteria use the human growth hormone gene to make human growth hormone, and I could just isolate just the protein if I wanted to. So you could do this for proteins other than human growth hormone. So this slide's showing how you could use it to make copies of a gene for pest resistance that you can put into plants. You could, over here, they're using a protein to make snow form at a higher temperature. So you can really use this technology for any protein you want. We're just talking about it in terms of human growth hormone for the treatment of dwarfism. Okay, so let's break this down a little bit. So here are just the steps that I just talked about with those two pictures. So first, we have to make the recombinant DNA molecule. Make the molecule that has DNA from two different sources in it. So what we do is we first isolate the gene we want, so the human growth hormone gene, and cut it with what we call a restriction enzyme. This is an enzyme that's going to leave that little single-stranded end. Then we're going to cut the vector, which is the plasmid from the bacteria, with the same restriction enzyme, so we have a similar single-stranded end. And then we're going to use our good friend DNA ligase. Remember, that was the glue that sealed those Okazaki fragments, the enzyme that sealed the Okazaki fragments up in DNA replication. So now I have one solid piece of DNA. Then we can put it back into the bacteria and, use it to either isolate the gene or isolate the protein, or do a whole bunch of different things we can do with it. So let's break this down into some more detailed steps. So remember, the first step, back on the previous slide, step A, is we have to isolate the gene I'm interested in and cut it out with that restriction enzyme, which you can think about as as DNA scissors. So in this case, we're interested in the human growth hormone gene. So let's talk about restriction enzymes. These are those enzymes that are going to cut the gene out. So restriction enzymes are actually found naturally in bacteria, but you might think why. And the reason is bacteria, they're infected by viruses. And remember, viruses work by injecting their own DNA and taking over the bacterial cell. They kind of hijack it. So bacteria have these enzymes that recognize the viral DNA. So the restriction enzymes are these little green things. They recognize that viral DNA and they cut it up. So scientists realize that, and what they've done is they have pure samples of these little proteins, these restriction enzymes, and they basically use them to cut up the DNA in the lab. So hundreds of different restriction enzymes have been purified and are available to order just through a catalog. And where they work is they recognize DNA, and what's cool about them is they cut at a very specific base sequence. And that base sequence is usually between 4 and 8 bases long. So for example, the restriction enzyme in this picture over here with this DNA, it's recognizing the sequence CTTAAG. So whenever it sees that sequence in the DNA, it's going to cut it. So let's look at, there are two types of restriction enzymes. There are ones that cut what's called a sticky end. So this is a really useful form of restriction enzyme. It's the more useful form. And EcoR1 is an example of a sticky end restriction enzyme. Remember, there are hundreds of different ones. EcoR1 is just one. So what it does, whenever it recognizes the sequence GAATTC, it makes a cut. It cuts right between the G and the A. Now remember, DNA is double-stranded and it's read 5' to 3', and then the other end goes 5 to 3. One of the really cool things about restriction enzymes is they only cut at palindromes. And if you remember from English class, probably in middle school, a palindrome is something that's read the same forward and backwards. So if you look at this DNA sequence, if you look 5 to 3 on the top strand, it's GAATTC. And then if we read 5 to 3 on the bottom strand, it's GAATTC. So it's the same sequence read forward and backwards. So this EcoR1 is going to recognize this GAATTC, cut between the G and the A, and also cut between the G and the A on the other strand, which basically leaves this strand where after it breaks, you can see I have a single-stranded overhang on one end, and I have a single-stranded overhang on the other end. Okay? So this is why it's called the sticky end, because now these single strands, if you put them together with other single strands cut by the same enzyme, they're going to want to stick together, because DNA does not like to be single-stranded. So that's why they're useful. The other type of restriction enzyme is called the blunt end cutter, and this is an enzyme that cuts right down the middle. There's going to be no overhand, no sticky end. But again, they're still cutting a palindrome. So SMA1 is another restriction enzyme that happens to be a blunt cutter, and it recognizes CCCGGG, going 5 to 3. So it's going to cut between the C and the G, and then in the other direction, CCCGGG is going to cut between that C and G. Okay, so these kind of enzymes are less useful because there's no single strand overhang on these ones, so they're not as useful for trying to combine different DNA molecules. Okay, so let's look at this in a little bit more detail. So maybe here's my chromosome, the whole chromosome that I'm looking at, and maybe my human growth hormone is right here. Okay, so if I take a restriction enzyme like EcoR1 we saw on the previous page that's going to cut anywhere it sees that 6-base sequence. So maybe there's the 6-base sequence right here, so it cuts the DNA right here. And that 6-base sequence is also here, and here, and here, and here. And maybe there's one right here, and one right here, and then one here. So this is going to cut my DNA into 1, 2, 3, 4, 5, 6, 7, 8, 9 pieces. So now I'll have 9 pieces of DNA, and one of my 9 has the gene I'm interested in. Okay, so I formed many restriction fragments. So restriction fragments, what we call a little piece of DNA, are formed, and they all have different genes inside of them. Okay, so now what we do is we put all of these fragments into what we call a vector. So what a vector is just anything that can carry our gene of interest. Okay, so there are mechanical vectors. We aren't going to talk about these much, but there are going to be things like micropipettes, so little bitty pipettes that you can actually use to inject genes into cells. Or gene guns, which are literally like high-powered guns that shoot DNA at high speeds into cells. We're going to be a lot more interested in biological vectors. So I could, for example, put my human gene of interest into a virus, or I can put it onto a plasmid. Remember that plasmids were those little tiny extra chromosomes found in bacteria. So now what we're going to do is, so we used a restriction enzyme to cut up my gene of interest. I'm going to use the same restriction enzyme now to cut up my vector. So I can take a plasmid, that additional free-floating ring of DNA found in bacteria. If I cut it with the same restriction enzyme, it will have the same sticky end as my gene of interest does. Okay? Plasmids are kind of cool. A couple notes. They can replicate on their own inside of bacteria. So they have an origin of replication, and they also have antibiotic-resistant genes. This is going to become important a little bit later on in a few videos from now, so just keep that in mind. Okay, so if I cut both my plasmid and my gene of interest with the same restriction enzymes, their sticky ends will match, and they'll be able to bond together. Okay, so I have a picture of that here. So you can see, this is my human growth hormone, my gene of interest. I cut it with a restriction enzyme, and now it has these single-stranded overhangs. If I cut my plasmid, remember this is a plasmid, so it's actually going to be, a circle. If I cut it with the same restriction enzyme, my gene of interest will fit right into that plasmid because the sticky ends will match. Okay? And then I use ligase to fill in these little gaps. And there I go. Now I have my human gene inside of bacterial DNA. So this is another picture that shows the same thing. I actually like this picture a lot more because it's actually showing the plasmid as a circle. So again, we're using the EcoR1, that enzyme from a few slides ago. We're using that enzyme to cut both the plasmid and my DNA of interest. It's going to cut in many spots. And then maybe this one right here has my human growth hormone, and these ones have other genes on them. Okay? So each one is going to go into a plasmid, and now I have a plasmid bacterial DNA holding my human gene. So that's it for video 1. We'll see you in video 2.